Okay, I make that just after the hour, so I think we'll get started. Good morning, good afternoon, good evening, wherever you're joining us from. In Japan, good night, I guess. And welcome to today's Twist Bioscience webinar, Decoding Transcriptional Regulation Using Massively Parallel Reporter Assays and Twist Oligopools. My name's Chris Thorne. I'm Senior Manager for Field Marketing here at Twist. So before we get into the webinar, I just want to do a little bit of housekeeping. As it is a webinar, you'll all be on mute as our audience participants, but there will be a Q&A after the webinar, and you can submit your questions using the Q&A box at the bottom of your Zoom webinar window. So please use the Q&A box rather than the chat box, and this will just help us manage those questions as they come in. Additionally, once the webinar finishes and we're done with the Q&A, if you do have a minute at the very end, we're going to forward you on to a brief survey, and we would appreciate your feedback. In fact, this is how we improve, so that would be much appreciated. And so it gives me great pleasure to introduce our speakers today, Dr. Cosmas Arnold and Bernardo Almeida, both of whom join us today from the Research Institute for Molecular Pathology in Vienna. Cosmas is a research associate in the lab of Dr. Alexander Stark, where he pursues his interests in gene regulation, functional genomics, and technology development. And during his scientific career, he has developed several novel technologies to study gene regulation, including stepsiq and stepsiq. Bernardo is currently working alongside Cosmas in Dr. Stark's lab, where he is completing his PhD using computational approaches to understand the cis regulatory information encoded in the sequence of transcriptional enhancers. And so with that, I'd like to turn over to Bernardo, who's going to begin, and I'll let him take it from here.
Thank you, Chris. Can you hear me? Everything good?
All good.
Perfect. Yeah. So hello, everyone. Thanks for the introduction, Chris. So today, Cosmas and I want to give you an overview of the work we are doing in our lab. And the different elements involved in transcriptional regulation that we are studying using this combination of massively parallel reported assays developed in the lab and twist oligopools. So I will start, and I want to start with this slide and with an interesting observation in biology, which is that all the information to generate all our different cell types during development is encoded in a single genome. That cells have to differentially interpret to express specific sets of genes that define their identity. And due to the importance of this genetic information, so perturbations in the sequence like mutations are often the cause of disease. And therefore, understanding the information encoded in our genome is fundamental to understand both human development and disease. And so the human genome actually doesn't code only for protein coding genes, which is only around 1.5%, but most of the genome, in addition to repetitive elements, contains non-coding elements that regulate when and where each of these genes should be expressed. And these non-coding regions are often the target of disease-associated variants. And in our genome, we have two key DNA regulatory elements, promoters and enhancers. So promoters are DNA elements where the transcription machinery and pole to assemble. And then enhancers are distal elements that enhance the transcription of target genes. And enhancers work by recruiting transcription factors that bind in a sequence-specific manner to the enhancer DNA, and then recruiting subsequently cofactors. So another set of proteins that communicate with the promoters to drive transcription. And in our lab, we are studying these different elements, both the sequence elements and the transcription factors and cofactors. And today we want to share with you two different projects. So I will start first with some unpublished data from a project where I teamed up with Francisca in the lab to study the information encoded in the DNA sequence of enhancers. And then Cosmas will follow with his joint work with Vanya on cofactor promoter compatibilities, a work that was published a couple of years ago. So starting by enhancers and coming back to this DNA sequence that I showed in the beginning. So in this region, actually there is a presence an enhancer, so a human enhancer that activates the sonic hedgehog gene, specifically in the limbs. And this exact DNA sequence that encodes for that enhancer is sufficient to drive transcription when isolated in reporter constructs, specifically in the limbs. So this piece of 800 base pairs is sufficient to drive enhancer activity and gene expression in specific cell types. And this is an important aspect of enhancer sequences, showing that the enhancer activity is actually dictated by its DNA sequence, and therefore we can study this piece of DNA outside of the genome context. And so, however, how to understand the information encoded in this piece of DNA is still very challenging, and one of the main challenges in biology. And this is the main question we are interested in. So what do we know about this cis-regulatory code? So we know that the cis-regulatory information is encoded in the sequence, as I showed, and these enhancer sequences usually contain motifs that are recognized by transcription factors, and the combination of these transcription factors drives enhancer activity. And so the current approaches to analyze enhancer sequences are based on identifying all motif instances in an enhancer sequence based on known transcription factor motifs, like the ETS motif has shown here. However, these current approaches usually cannot or usually don't validate or systematically test the contribution of these motifs to enhancer activity due to technical limitations. And therefore, we usually don't know which motifs are important, or also which, if different enhancers depend on different transcription factor motifs. And in addition, the motif syntax rules, so how these motifs are organized in the sequence, are also very elusive nowadays. And this is mainly due to technical limitations that we want to show you today how we are overcoming with twist oligopools. So in this project, to better understand enhancer sequences, we wanted to do a functional and systematic characterization of the contribution of transcription factor motifs to enhancer activity, combining in a powerful combination a massively parallel reporter assay developed in the lab by Cosmas called stepsiq that can quantify the activity of millions of sequences in parallel, and combine these with twist oligopools. So using the recent 300-mer oligopools, it allows us nowadays to synthesize enhancers and enhancer variants, and which we use to perform motif mutations in a systematic and large scale. So with the combination of these approaches, in this project, we want to address if different enhancers depend on different motif types, and also how the contribution of motifs depend on different motif syntax rules. So in our lab, we use the Drosophila enhancer from S2 cells as model system. And previously, we have performed stepsiq in these cells to identify all the enhancer genome-wide, but those stepsiq experiments were done with 600 base pair fragments. And nowadays, due to the size limitations of the synthesis and the oligopools, so in the beginning, we had to improve or to this stepsiq into higher resolution screens to identify shorter enhancers that will be active at a short length that we can use then for the oligopools. So the first thing we did was to improve stepsiq or to have a more high resolution stepsiq using a 200 base pair genome-wide library. So not yet using the oligopools, that will be later, but first just a shared genome-wide library. And used stepsiq to identify all genome-wide enhancers. And the way stepsiq works is by placing all candidate sequences, millions of candidate sequences downstream of the promoter, such that active enhancers will activate transcription, transcribe themselves, and their activity is reflected by their abundance in the mRNA. And so we performed this stepsiq genome-wide. And also before in the lab, so we found two main enhancer types, enhancer that activate the developmental promoter and enhancer that activate the housekeeping promoter. And to characterize both enhancer types, we performed stepsiq with either a developmental or a housekeeping promoter. And this is how the data looks like in a specific locus. So here I'm showing a genome-wide track where each peak is an enhancer, so a sequence that activated the promoter in the stepsiq and therefore gave rise to a lot of mRNAs with the same sequence. That's why you see a peak. And showing that genome-wide, we identified 12,000 developmental enhancers and 7,000 housekeeping enhancers. And as you can appreciate, they are non-overlapping, meaning that a sequence that can activate developmental enhancers usually cannot activate, a developmental promoter usually cannot activate the housekeeping promoter, and vice versa for housekeeping enhancers. So after having these sets of short developmental and housekeeping enhancers, we then characterize their motif composition by comparing the motif, the transcription factor motifs in their sequence with negative control regions in the genome, identifying transcription factor motifs enriched in developmental or housekeeping enhancers. And then we compared the two sets of motifs to see if developmental housekeeping motifs contain different or distinct motif types. So I'm comparing here the motifs enriched in developmental enhancers with the motifs enriched in housekeeping enhancers to show that they contain different motif types. So for example, developmental enhancers usually contain AP1 or GATA transcription factor motifs, while housekeeping enhancers are enriched in DREF motifs, OLR1 motifs, among others. And this really explains why the developmental enhancer can only activate a developmental promoter and not the housekeeping promoter, and vice versa, because they contain different proteins that are activating the promoter, and then due to the specificity, they can only activate specific promoters, as Cosmas will also talk later. So after having this set of candidate motifs, the goal was to really functionally characterize their contribution by perturbing them in the enhancer sequence, so performing a systematic motif mutagenesis. So we selected the 4 top developmental motifs and 3 housekeeping motifs, mapped all their instances in several strong developmental and housekeeping enhancers, and then designed computationally sequences where we mutate all instances of each motif type at a time, here shown in different colors. So we computationally designed these sequences and then used the 300-mer oligo library from Twist to synthesize these sequences. So we synthesize sequences with the enhancer variants, so enhancer sequences with the motifs mutated, as well as the wild type sequences. And then we use this combined library as input to the same stepsiq assay, now assessing or quantifying the activity of each wild type and motif mutant variants. And by comparing the wild type with the motif mutant for the same enhancer, we can really functionally characterize the contribution of that perturbed motif to enhancer activity. And here are the main results. So I'm showing the results with has the log 2-fold change by comparing the wild type with the motif mutant. So stronger log 2-fold change will mean that the motif, when mutated, reduce the activity of the enhancer, and therefore is important for these enhancers. And here I'm showing for developmental enhancers, the impact of mutating 4 control motifs, then the 4 predicted developmental motifs or the predicted housekeeping motifs. And here we are really talking about 5 or 6 base pair mutations. And this shows that only the developmental motifs are important for developmental enhancers, but not the control or the housekeeping motifs. And this is exactly the same experiment, but now in housekeeping enhancers, showing that now only or mostly the housekeeping motifs are important and not the developmental motifs, even if they are present in the sequence like these ones. So a very clear example is the distinction of AP1 that is very developmental specific, not important for housekeeping, while DREF, for example, is only important for housekeeping and not developmental enhancers. So this really shows that these 2 enhancer types not only have different motif composition, but those specific motif types are specifically required for either development or housekeeping enhancers. And with these oligo pools and the accuracy and the quantitativeness of stepsiq really allow us to discriminate log 2-fold change at a very high magnitudes. And this is only possible because of the dynamic range that we have in the stepsiq assay, that we can now see drops of log 2 of -2 or even -8 to -10 fold. And so in addition to these specifically requirements of these enhancer types, a very interesting observation was that for the same motif, we had enhancers that were very dependent on that motif, while others were less dependent. So you can see that these distributions are still very, very variable. And so, for example, for GATA, you have motifs that require less the GATA motif and others that really require GATA. So this suggested to us that in a sequence, there should be additional sequence features that explain why GATA is more important in some enhancers than in others. And this was true for all the other motifs. And indeed, so one feature that we found was very important to explain this variability was the number of instances of a motif that is present in the enhancer. So what do I mean by this? So here is exactly the same log2-fold change, but I'm dividing the developmental enhancers according to the number of gathers that they contain. So similar to this cartoon here. And as you can see, so with increasing number of gather instances, those enhancers became more dependent on that motif when we mutate the motif. And this is really independent of the wild type activity of the enhancers, which is the same for either 1, 2, 3, or more than 3 gathers, because we only selected strong enhancers. And this is also independent of the proportion of sequence that we are mutating, because when we did this for the control motifs, which is more difficult because they are more rare, but we didn't see this constant decrease in activity or in log2-fold change with increasing number of mutated instances. And this was true for gather, but was also true for all the other developmental motifs, as you can see here. And also in housekeeping enhancers, this was also true for the 3 housekeeping motifs. So showing that it's really a general feature, enhancers that have more instances of a certain motif are also more dependent on that motif. And that explains why the initial box plots were so large. We next compared these dependencies of a motif to the transcription factor binding endogenously. So here I have an example starting with the twist transcription factor. So I told you before that increased number of the twist motifs in the enhancer correlates with higher dependency of the enhancer, and this is independent of the wild type activity. And this correlates also endogenously with stronger twist binding. So higher number of twist instances also leads to or correlates with stronger twist binding. And this was similar, or this correlation was true also for the other transcription factors for which we had stepsiq data. So TRL also increasing with the number of instances, DREF in housekeeping enhancers, and M1BP in housekeeping enhancers. So in addition to these functional requirements being dependent on the number of instances, this is also correlated with stronger transcription factor binding. So suggesting that enhancers with more instances are more dependent on that transcription factor, and in opposite, enhancers that have less instances of a specific motif have usually a more diverse composition of different motif types, and therefore are not dependent so much on a single transcription factor. And so I want to finish now with the main highlights or the main results I showed you today. So we wanted to really functionally characterize the contribution of motifs to enhancer activity, and for this we started doing this high resolution stars to identify short enhancers that we could synthesize with twist oligopools. Then we characterized the motifs enriched in these enhancers and performed a systematic mutagenesis using the twist oligopools, really synthesizing sequences with just a 5 or 6 base pair variants. And these showed that developmental housekeeping motifs are specifically required for the respective enhancer types. And an additional and global feature that we found both in developmental and housekeeping enhancers was that enhancers with more instances of the same motif are more dependent on that motif, correlating also with stronger transcription factor binding. So to us, I think also this correlation between the endogenous transcription factor binding and the reported assays that we are doing really makes us confident that we can use this approach to study enhancer features. And also these oligopools are now allowing us to go to more complex motif syntax rules, perturbing the enhancer in different ways to test different hypotheses and combining them with stepsiq, I think in a very complementary way. So I want to finish here. Hand it over to Cosmas, and I'm happy to take questions in the end. Yeah, thank you very much.
Yeah, so thanks, Bernardo, for this really interesting talk about enhancers, and we switch now gears a little bit and talk about promoters. And as Bernardo already mentioned, this is a project I shared with Vanya Habele, a fantastic postdoc in the lab, and she basically did all the bioinformatics analysis, processing all data I'm going to show you now. And so I want to more specifically talk about how enhancers communicate with promoters in order to activate gene transcription from the promoter. And this communication is done by non-DNA binding proteins called transcriptional cofactors. And these transcriptional cofactors are a highly diverse set of proteins that contains, for example, histone acetyltransferases like the well-known P300 or MOF, bromodomain containing proteins like the famous BRD4, histone methyltransferases of the MLL type, for example, like LPT or TR, that's the Drosophila paralox, or of course the well-known mediator complex. Now, if we want to understand how enhancers regulate or activate transcription from promoters, we first need to understand how cofactors activate or regulate promoters, as they are basically in between and responsible for the communication. And given the sheer number of promoters in the genome, it is unclear whether every cofactor, like in a naive model, every cofactor can activate every promoter, or if there is some sort of specificity or compatibility, so that meaning that a specific cofactor only activates a specific promoter or types of promoters. And an ideal experiment to map out, and mapping out this compatibilities is the main goal of this project, to map out this cofactor promoter specificities, is of course to take a single cofactor and then ask which of the thousands of promoters in the genome can be activated by this single cofactor. And you might expect some picture like this, that some promoters cannot be activated, while other promoters can be weakly or even strongly activated by a given cofactor. And then, of course, to display the complexity of the cofactor proteins, we want to do this for several cofactors, where we then would find, for example, that promoters that cannot be activated by a given cofactor can now be activated by a set of different cofactors. And as Bernardo also already mentioned, so we use a massively parallel reporter assay called stepsiq that we developed in the lab to measure promoter activity. And for this, we clone a promoter candidate library at the position of the core promoter within the stepsiq reporter plasmid. And this promoter library is comprised of 72,000 candidates derived from twist oligo pool, and this basically contains every promoter in the Drosophila genome. And we then co-transfect this promoter library together with a single cofactor that is fused to the DNA binding domain of Gal4, so that we can that this cofactor is recruited in front of this promoter library. And if a cofactor can activate a specific promoter, this will give rise to reporter transcripts that start with a small sequence tag derived from its target promoter, and which informs us later about the identity of the promoter. We then capture those reporter transcripts by ligating 5' RNA linkers that importantly also contain a 10 nucleotide random barcode, a unique molecular identifier that allows us to count really individual molecules and quantify precisely the activity of the promoter in response to this cofactor. We then amplify those sequence tags and then map them back, deep sequence them, map them back to the genome, and quantify them as mentioned using this UMI-eyes. Now, as mentioned already, we don't want to do this for one cofactor, but we did this for 13 cofactor plus 2 controls, and to have enough statistical power, we did 3 biological replicates per cofactor, and all the work you will see now is done in Drosophila S2 cells. Now, this is how the data looks like. So I first show you now the controls here, where P65 is our positive control, and you can already appreciate here that P65 specifically activates those 2 promoters, while our negative control, where we recruit GFP, expectedly doesn't activate any of the promoters. So if we extend this now to more cofactors, For example, the histone acetyltransferase P300, we see it activates the same promoters that than P65, but another histone acetyltransferase MOF activates very different promoters in this locus. And for example, this ribosomal protein gene here, the promoter of this ribosomal protein gene can only be activated by this cofactor by chromator. And this already shows that different cofactors really activate different promoters and not all the same. And this also holds genome-wide. If we cluster all 13 cofactor screens plus the 2 controls by similarity, we find that some cofactors activate very similar promoters, and other cofactors activate very different promoters, establishing that there is like a widespread cofactor promoter specificity, probably conferred by some kind of biochemical compatibility. We then also could validate this using individual luciferase assays for 50 individual promoters. And on top, I show you the stepsiq signal of those promoters, and on bottom, the luciferase assays. And as you can see here, the agreement between the 2 assays is very high, proving or validating our stepsiq results. Now, everything what I've shown you so far is derived by bringing together on a plasmid a cofactor and a promoter. So the question is, how does the cell in vivo make use of this, and how is the endogenous situation? And for this, we rank our stepsiq signals for all the promoter candidates according to strength. And we see that the strongest activated promoters by, for example, P300 in our assay are also the ones which are bound the strongest in vivo according to ChIP-seq. And for this, we used publicly available data sets for this comparison. And we find a similar picture for BRD4 and TRX. Now, if we staying with this, with the top ranked promoters, if we ask, okay, what happens to the genes that are associated with those top ranked P300 promoters in vivo, once you inhibit P300, we find those genes the most downregulated ones compared to P300 independent genes or control genes. And again, the same is true for BRD4 and TRX. And vice versa, if we look into the most downregulated genes upon P300 inhibition, we see that those most downregulated genes are the ones which have the highest stepsiq signals in our screen. And again, this is true for BRD4 and TRX. So, and I think with this, we already have a good proof that this cofactor promoter compatibility is also indeed used by the cell to activate or to specifically activate certain genes. Now, now I think with this also we disproved this naive model where basically every cofactor can activate every promoter, and rather established that there's this widespread specificity. Now the big question is, of course, what is the basis of this specificity, and what's its implication? And for this, we switch now to a promoter-centric analysis, where we first score each cofactor's relative contribution per promoter, and then we cluster the promoters according to their relative activity profiles, leading to 5 distinct promoter groups, where group 1, for example, group 1 promoters are the strongest activated by P65, group 2 promoters are the strongest activated by MET25 or mediator complex, presumably. Group 3 is mainly activated by another histone acetyltransferase, MOF, and group 4 by chromator, and group 5 by GFCF. And again, here, I think it becomes very clear that a single promoter can be very differentially activated by all different cofactors. So now the question is, okay, what is the basis of this? And for this, we first looked into the sequence of those promoters, and most importantly, we looked for the well-known core promoter motifs. I think everyone should at least know Tata box, and we find that indeed group 1 and group 2 promoters are highly enriched for Tata box compared, for example, to group 3 to 5, where Tata box is completely absent, and also for initiator motif, while group 3 and 4 promoters are highly enriched for this OLA and DRE motifs. And I think this can be even seen better here in this enrichment analysis, where group 1 and 2 promoters are enriched in this, what we would call canonical core promoter motifs, while group 3 and 4 are enriched for this OLA, DRE, and EBOX motifs. And this, I think, clearly shows that this specificity is kind of encoded already in the promoter sequence. Now, sequence is not everything, so this is why we also looked into the chromatin structure of those promoters. And again, here we also find very distinct features of the different promoter types. So if we look into the chromatin accessibility, for example, then we see that group 1 and 2 promoters have a much broader region of accessible chromatin as compared to group 3 and 4 promoters, and this is also quantified globally here. And while on the other hand, we find that group 3 and 4 promoters have very precisely positioned or phased nucleosomes, which is not the case for group 1 and 2 promoters. And then, of course, the big question, what's happening to the histone marks? And just to remind you, H3K4 trimethylation has in literature be the canonical promoter mark for that marks active promoter. And to our surprise, we find that for group 3 and 4 promoters, I mean, it's the expected picture, you have high levels of K4 trimethylation, but group 1 and 2 promoters seem to be much less K4 trimethylated. And the biggest surprise then came when we looked into K4 monomethylation, and K4 monomethylation is considered as an enhancer mark. However, the group 1 and 2 promoters seem to have high signals for K4 monomethylation. So is this really that the genome has now a lot of K4 monomethylated promoters? And yes, there's many instances of this. And just to illustrate this to you, I show you one screenshot here. So we have those two genes are both active endogenously. This here is activated specifically or by P300, has high levels of K4 monomethylation and no K4 trimethylation, while this gene here is activated by MOF, has high levels of K4 trimethylation, but K4 monomethylation is absent. And our data provide, and if we look into the gene ontology of those genes, then we find, as Bernardo already introduced at the beginning, we really find that group 1 and 2 promoters are associated with mainly developmental genes, and group 3 and 4 promoters are associated with housekeeping genes. And our data provide even more cool instances of differential regulation, where, for example, there are several instances where like within the same genes, different isoforms, as shown here, are regulated by different cofactors, in this case P300 versus MOF. And even within the same promoter, different cofactors use different transcription start sites, as shown here. Yeah, and I think taken together, we basically provide a pretty complex picture, suggesting that there is extensive cofactor promoter specificity. And as Bernardo already mentioned, we previously found that there's already enhancer core promoter specificity extensively, separating basically housekeeping and developmental gene regulation. And I think with this story, the loop closes, because it's the transcriptional cofactors that confer this specificity. Now, as mentioned, all what I showed you now was done in Drosophila S2 cells. And of course, the burning question is what happens in mammalian cells or in human cells. And for this, we also performed stepsiq in human cells, in human colon cancer cells, HCT116 cells. We didn't include all promoters. We used what we call focused library that's comprised of 12,000 candidates, which is a representative snapshot of all promoters in the human genome and contains about 10% of those promoters. We screened 5 cofactors and also controls, and again did 3 biological replicates. And as you can appreciate from those 2 screenshots here, we find a similar picture as in Drosophila, so that this gene, for example, can be activated, or the promoter of this gene can be activated by P65 or MET15, while this CpG island containing gene IRAK1 is mainly activated, or is strongest activated, by MLL3. And if we now cluster all the screens we have done in human cells, we also find a similar picture compared to the fly cells, where some cofactors activate very similar sets of genes and other cofactors activate different sets of genes. And as a last piece of data, I'm going to show you, we also, of course, digged a little bit into the sequence and compared MET15 preferred promoters versus MLL3 preferred promoters, and we find that they are differentially enriched for TA or GC dinucleotides, pointing already to kind of a Tata box and CpG island promoters maybe. And indeed, if we look into the Tata box, we find MET15 preferred promoters highly enriched for Tata boxes, not so much for initiator. And vice versa, the MLL3 preferred promoter have a much higher GC content and also much more CpG dinucleotides. And this basically shows that also in humans, a similar mechanism is used, and also Tata box versus CpG island promoters is regulated by different cofactors. Yeah, and with this, I also would like to end and give you a quick summary. So we established to map out cofactor promoter compatibilities or specificities throughout the Drosophila genome and for parts of the human genome using our massively parallel reporter assay stepsiq, coupled to twist oligo pools to synthesize our promoter library. We find extensive cofactor promoter specificities, probably due to this cofactor or to a biochemical compatibility between cofactors and the promoters, and also have a hint what might confer the specificity between enhancers and certain types of promoters. We find that this is all or partially sequence encoded, and that the different types of promoters we were able to define also differ in their chromatin features or chromatin structure. And this is kind of the model we would propose. So cofactors can only activate very specific types of promoters, and even within the same gene, different isoforms can be activated by different cofactors, and even within a same promoter, different cofactors can make use of different transcription start sites. And with this, I would like to thank a lot of people. First of all, of course, Vanya, as mentioned already, she did all the bioinformatics analysis, and we basically headed this project, and we're also co-first authors on this publication. Of course, Alex Stark, our boss, who mentored us through this entire project and supervised the whole thing, and then our fantastic technician team, Michi Pagani, Martina Rath, and Kati Bergauer. And to mention Michi, because I think every single piece of data you saw, she basically had her hands in the wet lab on them. And then, of course, the entire Stark lab. And I also want to thank Bernardo for this fantastic presentation on enhancers, and also Fanny for doing the wet lab work on this. And I also really need to mention our amazing core facilities here on campus at IMP IMBA in Vienna. And also, of course, the next generation facility at the Vienna Biocenter core facilities. And of course, our generous funding. And I will end with thanking you for attending this seminar.
So thank you, everyone. And I look forward to seeing you at the next webinar. And bye for now.
Hey, thanks, everyone. Super cool. Thanks.